Hello and welcome to this episode of Our Longevity Futures. Now today's talk is from an as of yet unpublished interview that I held back in the summer of 2020 with the husband and wife team of Professor Michael and Professor Irina Conboy, both of whom are professors at the University of California, Berkeley, in the Department of Bioengineering. Now back in 2005, their discovery of the rejuvenating effects of young blood through parabiosis in a seminal paper published in Nature paved the way for a thriving field of rejuvenation biology. The Conboy lab currently focuses on the broad rejuvenation of tissue maintenance and repair, stem cell niche engineering, elucidating the mechanics underlying muscle stem cell aging, and making CRISPR a therapeutic reality. In this talk, we discussed their fascinating paper titled Diluting Blood Plasma Rejuvenates Tissue and Reverses Aging in Mice. So I really hope you enjoy it. Before getting into the most recent paper, could you just give me an overview of what your 2005 parabiosis study was? And what have we learned since then concerning the relationship between aging and the blood?
All right, yes. So our 2005 study was really focused on hypothesis that aging could be reversed in old mammals rather quickly. Because before that, people thought that once you have shortened telomeres, accumulated DNA damage, genetic drift, then it is a single destination train which will not go back. So that was the hypothesis of 2005 work, which Mike and I started exploring experimentally back in 2002. And Mike also had this idea that because we age synchronously in various parts of our body, the system that connects those parts is bloodstream or circulation. So therefore, we wanted to look at the effects of circulation. The goal of the study never was to prove that young blood could work as a medicine. And to our opinion, it never was concluded or conclusive from the outcomes.
So could you tell me the results of your recent study?
Sure. So let me do a quick bridge between the parabiosis, right? So the parabiosis is like a Siamese twinning of the young and the old animal. And they share blood, but they also, because they're connected for a very long time, a relatively long time, they share the tissue and organ systems of the partner. So the young animal suffers half its mass or more than half its mass being old and having old liver, old kidneys, blah, blah, blah. The old partner gets access to a young set of liver and kidneys and pancreas and insulin regulation and digestive system and whatever. So we didn't know for sure if it was the effects that we saw were because of the blood or because of the other systems that kind of communicate through the blood. The next experiment we did was a blood exchange. So we just took two animals and transfused blood back and forth between them. And then after several exchanges, you can approach homogeneity between them. So there's about 50-50, and then disconnect them. And because the only thing that was transferred was the blood, we could rule out the effects of organs and regulatory systems. So from that work, we found that the liver was improved in the old animal. The muscle regeneration was improved. The brain wasn't really all that improved. And the young partner that got old blood, the muscle regeneration was a little worse. The liver couldn't really see much of a defect for the things that we looked at, but their brains looked like they got stupider. So then at the end of that, we said, okay, now did this happen because the old animal got an infusion of young factors, young pro-youth factors, or did it see improvement because it got rid of half of its old bad factors? And conversely, the young animal, did it get worse because it got these old factors or because it got rid of half of its good factors? So what if we could do a blood exchange like we did between the young and the old animals, but with each of them, have them exchange with something that was neutral? And we tried to figure out what age of mouse could be neutral for, but it's difficult to point to a particular age. So we said, what if we just hypothesize, say, okay, let's say it's not the red blood cells and it's not the white blood cells, and that leaves plasma. So what if we had some blood that we throw out the plasma and replace it with purified saline and purified albumin, which is like the major ingredient in plasma, but it doesn't have any factors in it. It's just devoid of factors. It doesn't have any old factors, doesn't have any young factors. And do the exchange with that, would the old animal look a little younger and would the young animal look a little older? So that was the basis for the experiment that we did. So we took old mouse blood and pelleted the red blood cells and the white blood cells and threw away the platelet-rich plasma, right? So it's got albumin in there, it's got antibodies in there, it's got platelets for clotting, and it's got all the old factors that would be in old blood. And replaced that with saline, mixed in with the major purified component of blood, is albumin. And then did the exchange with the old mouse with that, and we did some with young mice too, so that in the end, the old animal has all its red blood cells and has all its white blood cells, and has half of its platelets, and has its plasma diluted in half by this purified plasma, basically. So that was technically what the experiment is. And then what we found was surprising. I didn't expect the old animal to be really all that much improved. I figured some things might change, but in other ways it'd get worse. But it didn't seem to get worse in pretty much everything we looked at.
So, as you said in this study, half the plasma of the mouse is replaced. Can you explain how this was actually done and what kind of controls you had set up, and how this might be scaled up to humans potentially in the future?
Yeah, so it's almost easier to work from humans back, right? If you've ever given blood, they stick a line in your arm, a needle in a major vein, and blood comes out and flows into this bag, and when the bag's full, they unplug you and you're done. Sometimes they say, hey, come back and we'll do apheresis and we'll collect platelets. Or they'll say, we'll get a double batch of red blood cells. For that, what they do is they put two lines in, and the blood comes out and goes into this machine that separates the blood. So you get the red blood cells going one direction, the white blood cells going another direction, the platelets going in the third direction, and the plasma going in the fourth direction. And then they can recombine all that on the other end to come back in to give you back 3 out of the 4. Or they can also infuse stuff in. So when you're donating platelets, the platelets go off into one bag, and everything else, the red blood cells, the white blood cells, and the plasma, comes back into you. And that way they can take a lot more platelets than they could from just 1 unit volume of blood. And this has been around for decades, right? And they also use the same technology for people that have, let's say, an autoimmune disease, right? So their blood is full of an antibody that's autoreactive against some of their own tissue, and they're going to die from that. So they run the blood through this type of technology, and then they do the same thing. They pull out the plasma that has the antibodies in it and throw it away, and replace it with a saline solution that has albumin. Right. Some people have organ transplant, and they're going to reject the organ. So they do a similar kind of thing. They pull out the parts of the antibodies, and there's other parts of the immune system too that will react against the donor's tissue. So they can pull that stuff out and separate it, but give the patient back their own red blood cells, their own white blood cells. And because you're not really getting anything foreign from somebody, it's just some salt solution and purified protein. Your own body's immune system doesn't usually react to it. So that's easy enough to do with people. With mice, you can't access like some vein on their arm because it's super, super tiny. Their veins start like where our capillaries end almost. Not really, because everything's governed by the size of a red blood cell, right? But they're very small. So technically what we have to do is we have to access the biggest vein we could find, which is the mouse jugular vein. And you stick a tube in there, and then from that tube, then you can pump in and out small amounts of fluid or blood or whatever. So that's technically how it was done. And so we had our old mice that got exchanged with other old mice. That's a control. And old mice that get exchanged with this neutral age blood made from purified albumin and saline. And then we also did as control young mice that were exchanged with other young mice. Right? And of course, in previous work, we had done those controls and then young mice that were exchanged with old mice.
So you say in your paper that the results do not exclude the possibility of beneficial factors in young blood having a rejuvenating effect. Is it theoretically easier to remove or neutralize harmful factors than to identify and introduce beneficial ones? And does this research bring us closer to an effective plasma therapies in humans?
Next segment.
Identifying factors, whether they're good young factors or bad old factors, I think is equally difficult. But if you're old and you want to get your young factors by getting units of young blood or young plasma, that has problems. And the problem is that the host immune system can react against the graft, the donor blood, and vice versa. The donor blood can react against the host. And once this reaction happens, then everybody's immune system starts participating and it can go to hell in a handbasket pretty quickly. I'm not an MD and I'm not necessarily an expert on this, but a little bit of reading I've done makes me think that the risks, the serious risks that you get from receiving a unit of blood product, whether it's plasma or whole blood, is on the order of about 1% per unit. So if you're getting a liter, that's 4 units. If you're getting 2 liters, that's 8 units. So we're getting close to about 10% risk for some people that are getting a lot of this young blood product. Whereas if you take your old blood and you run it through one of these apheresis machines, the risk for a full replacement of plasma is altogether on the order of 1% or less, right? So the risks are much, much less to not get someone else's potentially reactive blood, but to try to clean up the blood that you have. But like you said, ultimately, I think it's going to come down to identifying the factors, right? What are the factors that are in old blood that we need to neutralize, and what are the factors that are in young blood that we could add back? In which case, then the risks probably drop even lower than that.
And how does your study compare to, say, Dr. Harold Katcher's recent publication about the plasma fractions causing significant rejuvenation in middle-aged rats?
I'm going to make it a point not to comment too much on it.
Of course.
It appears to be a preprint or a manuscript that's just released for comment, because it hasn't really gone through the peer review process. Generally, I could say that it's not just him. There's other people that have looked at young blood, and we've looked at factors that are in young blood or old blood.
Like
oxytocin, right? Others have looked at for a long time fibroblast growth factor. You can name, pretty much.
And we also look
blank letter GF, right? Any growth factor.
Maybe. So now I can. There are numerous approaches published by others and us that demonstrated multiple tissue rejuvenation. Oxytocin, we published in Nature Communications. It's cited, I think, 100 times or more, 200 times. There is factors that are produced by human embryonic stem cells, in which we published, I think, 4 papers. And there is Bay Area biotech called Juvena. So I'm on scientific advisory board and a stockholder. So I would like to disclose. We publish on specific factors. So there are, of course, numerous proteins that could work in a limited fashion to rejuvenate tissues.
So obviously with some of the aging studies, they use epigenetic clocks to determine the aging of the subjects. Is this something that you've looked at, or I was just wondering what your thoughts were on that as a biomarker of aging?
So of course, others and us again look numerously at changes in epigenetics, which is, or you may know, it is not something in adults that is programming or reprogramming. We are not programmed or reprogrammed once we are adults, right? Otherwise, we would be in trouble. So rather, for example, in our work, in most recent work, we show a comparison in 500 or 300 proteins and how the levels changed. That, of course, means that if proteins are elevated, as we see, for example, youthful factor becomes now higher after neutral blood exchange or therapeutic plasma exchange, it means that the gene expression is reset to a different level, as we model in figure 6. And when there is more transcription, there is a more openness of the epigenetic locus for that gene, which is again is a textbook information. It is neither nor something that we discovered. When we looked specifically some of rejuvenative treatments, for example, we published on stem cells in 2015. If you look specifically at trimethylated histone H3, K4 versus K27, we do see resetting to younger state. However, we do not look at what is called clock. So we do not look at those specific loci which people implicate in chronological age. We are establishing collaborations to do that. But if you simply are asking, is epigenetic status rejuvenated? Of course it is. You don't have to look every time. We know that if RNA polymerase II is bound to gene promoter, then methyl binding proteins cannot bind, DNMT cannot bind, and then the locus will remain open. If there is less transcriptional factors, some call them pioneer transcriptional factors, then there is less RNA polymerase II, and then, of course, all of the methylation promoted changes take place. So it is textbook biochemistry.
Anytime you have a change in tissue state or cell gene expression state, you get epigenetic change.
Right. Exactly.
So just the process of seeing a rejuvenation of a particular tissue, you're going to have an epigenetic change.
Right.
So.
And I don't know why people try now, what is surprising to me, that people try to separate them and say, oh, you see rejuvenation and proteome rejuvenation, but is it also actual rejuvenation? If you look at any diagram in cell and molecular biology textbook, those are just different levels of regulation of gene expression. They are not separate from each other.
The scientific shock would be someone who sees a change, a rejuvenation or a change in tissue state, and doesn't see the epigenetic.
Exactly. That would be
novel.
That would be novel. And like put that point there, because it's not really our discovery. It is something that is textbook. Undergraduate students learn it in Bio 1A in first year at Berkeley when they are 17 or 18 years old. And so it is important that we do not forget that point. As Mike said, it would be surprising that if you observe reduction of fibrosis, inflammation, reduction of SASP, senescent cell associated secretome, you do not see corresponding epigenetic change. Yeah, and it is important perhaps to remind people, particularly broad.
I mean, as a diagnostic tool or an investigative tool, looking at the epigenome may not be a bad idea. There's we look at transcriptome and then often compare that to the proteome, so you can see what genes are turned on and do they make proteins and does that accumulate, right? And having yet another tool looking at the epigenetic marks on the chromatin is, certainly valuable. And in some cases, maybe will be easier to get different information or better information than you would from the other methods.
Sure.
Yeah, we're working on that.
And it seems to be that the elevated factors in old blood is maybe more the culprit for aging. What is dumping these factors into the blood? Like, where are they coming from?
To link it to another, I think, still very viable theory of aging is they call this inflammaging, right? So you have more inflammation in the body. And a lot of these inflammatory molecules, they're not just used by the immune system. A lot of them influence other tissues too. The immune system uses them to differentiate the immune cells into their final activated and infection fighting state. But other tissues will use the same molecules or very similar molecules and same signaling pathways to differentiate their respective tissues. We see that in muscle, we see that in neural tissue, and so on and so forth. So those factors tend to accumulate with age. And there's a lot of overlap, I think, between what we found elevated in the old blood and what seems to be reduced or signaled to be reduced after a round of this neutral blood exchange or therapeutic plasma exchange. So in a short statement, I guess that would be the kind of the running model that we're working on.
And just finally, one last question. So we often have people that want to experiment on themselves. They're sometimes called the biohackers. If they've read your paper and they've looked into it, what would you suggest for people who will try to self-experiment to make themselves younger by your method? Would you warn them against it, or say, wait a bit?
Technically, this isn't as easy as just drawing a unit of blood. Technically, you need not just the equipment, you need the high quality reagents, replacement fluid. We've seen a facility that does this, and one of our collaborator, Kiprov, has been doing this for decades. It takes some serious skill, right? A lot of the risks for them, they're not graft versus host, they're not immune autoreactive kind of stuff, but they often, I guess, like thrombotic. Someone throws a clot or the line is clogged or some technical thing like that you don't want an amateur being responsible about. I guess for biohackers, I would say wait, right? If you have a medical condition, you have access to a doctor and they agree to give you to do this, it's FDA approved. We know the doctors that do this know the risks, should know the risks, and I guess they could discuss it with them. That was the whole point of linking it to something that's FDA approved, is that it's not necessarily that it's a shorter path to actually turn this into a therapy. For biohackers, I'd say wait on this a little bit, whatever. People can give blood. They can give a unit of blood or give some platelets or something like that or whatever and donate some plasma. We know the blood banks know what the risks are and how often too much donation is too much. And they monitor your hematocrit and whatever, that kind of stuff. Yeah, go ahead and do that. That's fine. Right? You feel good about yourself. You get some cookies, right? Don't overdo it. They have limits on that, but you'll probably feel better. You'll definitely feel better about yourself and you get a cookie out of it. So you help somebody.
It's a win-win. So thanks again to both Irina and Michael for what was a really fascinating talk. And you can go and check out their work at the Conboy Lab website, where you can keep up to date with everything that they're doing. So thanks again, I really hope you enjoyed it, and see you again next time.